Well, good morning, class. Here we go again. The end is in sight for you all. I'm sure you're counting the hours. But let's try and focus back on the topic we're trying to get across to you. That is the rapid expansion in science, particularly in microbiology, which has then given us all these tools that have let us get into the realms of biotechnology, which is kind of where we're leading up to right now. You'll remember we talked last day about the amazing numerical importance of microbes, particularly the bacteria. I gave you that wonderful wow fact that if you took all the bacteria on the planet, you could cover France to a depth of 3 feet. Then we talked about the remarkable diversity that you find within the microbes. We talked about the different kinds, the viruses, the bacteria, the algae, the protozoa, all different kinds of single cells, which is what makes them microbes. We kind of zeroed in on the bacteria because those are the organisms, because of their amazing diversity, that have given us most of the tools that have enabled us to use them, to use either their products or to use the bacteria themselves to do things for us. And that's what biotechnology is all about, making money from microbes, but also hopefully making wise choices. And that's where the ethics comes in, and where you will all have control in the future. We talked about bacteria because we had to get across to you this concept of this piece of DNA that bacteria have, or some bacteria have, in addition to their large circular chromosome. And of course, you'll all remember, you're all saying it inwardly, I know you've been studying this, they all have this piece of DNA, a small ring called a plasmid. And that's very important because it doesn't contain many genes. That's the areas of DNA that would code for a protein. But often on these bacteria, it has important genes that help the bacteria survive. And survival and outcompeting your neighbors is what it's all about, because that's what makes that kind of bacterium dominant when it is challenged. And so the plasmid would often contain genes, for instance, to break down antibiotics, so that when you're treated with antibiotics, that bacteria doesn't mind. It continues to grow because it has a gene that produces a protein that can break that antibiotic and make it not effective. When you're thinking plasmids, be thinking about how that makes them very capable of sharing that plasmid with another bacterium. And a plasmid can come in and out of a cell very easily and can go into another bacterium. So you can have antibiotic resistance in one type of bacteria, and then by pure chance, that plasmid goes into a completely different kind of bacterium, and suddenly that one has antibiotic resistance. So some of you have been reading in the news as we go through different epidemics of this antibiotic resistance that can pop up, particularly in hospitals where they're constantly being challenged by antibiotics. Different kinds of bacteria suddenly become antibiotic resistant. And that is usually because they've acquired a plasmid. These plasmids move around in nature. But they also move around if we want them to move around, and that is why they're very good as vectors. That's the other word that you should have taken away from last week, because a vector allows us to put a gene into an organism we want to change. Okay? This should be making some sense, and you should also be reading those 10 articles or so that are on Moodle. Short, very easy to read, 31 pages in total. The examination on the 25th, 20 multiple choice questions, easy, covering what we talk about in here and those 31 pages of reading. Clear, everybody? I know some of you are starting to panic about passing the class. Okay, back to where we left off last day. The second kind of vector we were trying to introduce to you was known as the viral vector. So we wanted to tell you a little bit about what a virus was. Remember, it's not an organism. It's only a piece of DNA wrapped up in a protein. But they're very, very important. Think about this. Every organism on the planet has at least one virus that will attack it. We have hundreds of viruses that can attack our bodies. Every organism, whether it's a bacterium or whether it's a horse or a rhododendron, has viruses that can attack it. So if you do a little bit of logical thinking, you're all saying to yourself, there are more viruses on the planet than any other species of organism living, and you'd be correct. Huge, huge, huge numbers of viruses, all different and usually very specific for one kind of organism. So we can use them and be aware of them because they can go in and move pieces of DNA around. So let's just quickly review before we even try to understand that horrible diagram which I pulled off the web about how a virus replicates. So we'll just describe it first and then we can point to it on this diagram. It's not difficult. So you've got a cell, along comes a virus. The protein coat on the outside of the virus helps it recognize the correct cell that it wants to attack. So it sits on the outside of the cell, and then it injects its little piece of DNA into the cell. At that point, this DNA incorporates itself into the host DNA, and then it tells that cell to start making more and more viruses. As we said last day, once the cell is now full of viruses, they have to get out, and so they burst open the cell, which is a process called lysis, and then off go those hundreds of viruses in search of another cell to attack and take over. So that's why we call it the lytic cell, because it's lytic coming from lysis. And in form here, we can see that we have a bacterial cell. Remember, viruses attack every kind of living cell, so many of them will go into bacteria as well as into higher organisms. In this case, this simple diagram, it's a bacterial cell with its one piece of DNA in the chromosome. We've got a virus going in here and putting its DNA into the cell. It then starts to manufacture new viruses. These are assembled, and then the cell bursts open, and out come these viruses looking for a new host to invade. So, as we said last day, they're a nasty piece of work. So, just to put it into words, some viruses can insert themselves so that's the lytic cycle, but we're now going to talk about a second cycle, which is called the latent cycle. And that's when, again, it's probably easier just to think about it, and then you can look at the actual explanation there. Some viruses will insert themselves into the cell, but instead of taking over the cell's machinery right away, what they will do is just incorporate themselves into the host DNA. So in other words, let's assume that you have a virus that comes into your body and it invades one of your cells. It just hides in there and joins up to your DNA, your genome as it's called. And that's a clever trick, because that virus then just sits there, and then when your cell divides and divides its DNA, guess what? The virus is replicated as well. So you have this virus that sits inside you and divides and divides and divides as your cells grow. And then at some point, for reasons we don't understand, the virus suddenly takes over your cell's machinery, makes lots and lots of copies of itself, and bursts open your cell in the classic lytic cycle. So, hope this doesn't sound horribly confusing. There is a reason for going into this amount of detail, but all we're saying is a virus goes in, it can either take over your cell's machinery immediately and destroy your cells and replicate itself in a lytic cycle, or alternatively, it can go in, it can sit in your DNA, can sit there for months and divide and make copies, and then suddenly, through some trigger that we don't understand, it suddenly goes through the lytic cycle, makes hundreds of copies, and bursts open your cells in a nice classic disease. So, think about it. Cold sore, herpes virus. You can never get rid of a cold sore because that's a classic type of virus that inserts itself into your DNA. If you have a cold sore or are prone to cold sores, that virus is sitting about in cells at the base of your neck. And it's sitting in your DNA and it's replicating and you can't get rid of it. And then every now and then an environmental trigger comes along. Could be a piece of hot pizza that you burn your lips with. Some people it's with sunshine. Some people it's just because they get the cold. Whatever the trigger is, it makes that DNA from your neck cells migrate into your lip cells, and then that virus goes through the lytic cycle. It bursts open your cells. You have a visible cold sore. It releases viruses to try and find some other people's lips, and then it retreats back into your neck cells. And that's why we call herpes the name herpes, because it's Greek for creeping. This is a virus that creeps along through your body, and every now and then goes through the lytic cycle and then creeps back again. So there's no cure for it. It's incorporated in your DNA. Anyone else know of a latent virus we hear about a lot? If you've got chicken pox or had chicken pox as a child, you have still got that virus living in your DNA. If you're unlucky, as you get older, it will get a trigger, it will come out, go through a lytic cycle, and what's the disease? Perfect. So you see, viruses are a nasty piece of work. So reading off this, some viruses can insert themselves into DNA, and those are known as latent viruses like herpes and like the virus for chickenpox. And then as your DNA replicates, your cells divide, they replicate with it, and that's known as lysogenic replication. Okay, so what this means is, when a virus goes into your DNA, that's a vector. Because if we attached a gene to that virus, it would be able to then insert itself into a piece of DNA we want to incorporate that gene in. So think about it. A vector is a way of moving the piece of DNA around that we want. And that's why viruses are very important. But there's another problem with viruses, and this is a problem for the ethics of biotechnology. Think about a virus going in and infecting a bacterial cell or whatever cell you like. Viruses, when they come back out of the DNA, can be quite clumsy, and they can pull some pieces of host DNA with them. In fact, sometimes they can pull large pieces of DNA with them, and they could even pull out a gene that you want to keep in that organism. And if they pull out a gene and then go through a lytic cycle, they're making copies of that gene, and then they go and infect another organism. So think about it. If we were to release some super degrading, oil degrading bacterium, for instance, into the environment with a set of genes that make it really good at breaking down oil, there's a chance a virus could come along and switch that gene into another organism. A totally unintended consequence. That process is known as viral mediated transfer. So a virus has the ability to insert DNA, and when they leave, they can take extra pieces with them. It could be a gene of a useful product. So that's great, because this is what we want to happen when we control it. But think about it, this can also happen in nature. And that's something we would not want to happen, because we have no idea what the consequences would be if that gene starts getting moved around populations that we did not intend to have it. Transduction is the process by which DNA is transferred from one bacterium to another by a virus. So it's the process we use when we want to do it deliberately, but the point I'm trying to get across to you is we can also have this happen in nature. So at this point in time, there is a complete block on letting anyone release genetically engineered bacteria into the environment. We can use genetically engineered crops. That's allowed because the crop stays there in the field. But with microbes, you have no idea what can happen if that gene that you've put into do a specific task like break down contaminants starts moving around into other populations. This just really underscores that we don't understand anything about the natural world yet, and that's what's putting the brakes on this. How many of you here are scientists? Stick up your paw. One or two. And some are asleep, so you're not going to do it, or you're shy. Most of us that are scientists understand science. You know, chemistry, you add A and B together and it goes boom, or it goes bright red in color. In other words, there's predictability. All the sciences have predictability. You do this and that, and something will happen, and you know what it is. Think about it, that one branch of science which we call ecology, which is about understanding the natural world. We don't have any rules whatsoever in ecology. If I take an organism from this pond and put it into this pond, we have no idea what will happen. And that's the scary thing when you start playing around with manipulated organisms that can get out and go places you never intended them to go. It could really end up in a very big ecological disaster. And we'll touch on that next day. But that's the danger of viral mediated transfer, and that's why we have to be very careful with it. So, moving on to where this was all heading, you should have in your minds several words, if nothing else. Vectors can be plasmids, or they can be viruses, and this allows us to move genes around in the laboratory, and that is known as genetic engineering. It's the purposeful manipulation of genetic material to alter the characteristics of an organism. Other words that you might come upon when you're reading those popular articles: the genome is simply the complement of all the genes in a cell. So your genome is every gene you have in your body that makes up all your chromosomes of every cell. And the chromosome, as I just said, is where the DNA molecules reside, which are all associated into sequences which make up the gene. Okay, this is probably, okay, yes, just more revision. So in genetic engineering, is also known as recombinant DNA technology. Just think about it, don't let these words confuse you, and this is everyday literature, so be clear where it's coming from. It's the same thing. You can manipulate or engineer material by moving genes around, and that's just the same as recombining DNA. Recombining or recombinant DNA technology. They're the same thing. When you read them, they're the same. And then, as we've said several times here, because we want to get it into your brains, the vector is what you use to move the gene. And you need something to cut the DNA, like a pair of scissors at the molecular level. So if you're going to move a gene, you have to be able to cut it out. And we do that with something called an endonuclease. That just means that it's an enzyme, because it ends in ase. Remember, that's the rule. It's a nuclease, meaning it cuts nuclear material, so it cuts DNA, and the endo bit means it's inside a cell. So what that's telling us is there's an endonuclease that we can use to cut DNA. And that endonuclease comes from inside a cell where it normally cuts DNA. This is where I wish we could be interactive, but I want you all to think carefully. This is critical thinking. This is putting all the pieces together. Why would a cell make an endonuclease? Think about it. So, I'll give you a hint. It's a bacterial cell. Once again, bacteria are giving us all the tools for this biotechnology. So what we have here is a bacterium that's making an enzyme to chop down DNA. Why would a bacterium do that? I'm not really expecting an answer, but I'd like to think your wheels are turning and you're thinking about what that bacteria might have a need for endonucleases. You have all the information from what I've given you. This is classic logic, critical thinking. What would it use that endonuclease to kill? Correct. A virus. It's its only defense against viral attack. Think about it. A bacterium gets attacked by a virus. The virus injects its DNA into the cell. If it has an enzyme that can break down DNA, it might just survive that attack. They don't have antibiotics, or they don't have immune systems like we have. A bacterium just has an enzyme that hopefully can chop down that virus before it can attack it. So a lot of bacteria have endonucleases. There are many, many on the market. You can buy them so that you can use them as molecular scissors in the laboratory. It's getting into too much detail probably, but some of them cut and leave overlapping ends, which are known as sticky ends, and that just helps it to stick into the new plasmid that you're inserting it. So think about it, a plasmid, cut it with some scissors, leave sticky ends, and the gene comes along and glues itself onto those sticky ends. It's as much detail as you need to know. Here it is in diagrammatic form, and this was the very first use of recombinant DNA technology. It's now probably 20 years old. The company that did this trick was Eli Lilly. It used to be that anyone that had insulin got it from, do you know where insulin comes from normally? Where do you find insulin in the body? Quite correct, the pancreas. And so they used to harvest insulin from pig pancreas. So it was a very expensive, lengthy procedure where they literally had to process lots of pig pancreas to make this drug that so many people have a need for if they have diabetes. So how nice would it be if you could take a piece of pancreas, cut out the gene that makes insulin using some restriction endonucleases, stick it into a plasmid, stick it into a bacterium that grows fast, and guess what? That bacterium will start to make insulin. It's got the gene and it will make insulin. And that's what they did. They chose the bacterium Escherichia coli that we talked about last day because it grows very fast. You will all remember that one E. coli can divide in 15 minutes, meaning in 12 hours. It gives you a population of 1 billion E. coli. And if we put a plasmid into E. coli and grow it in a big fermenter, we don't release it, we just grow it in a big laboratory, then every one of those E. coli is making insulin and releasing it into the fermenter where it can be harvested. So about 98% of all the insulin in the world now is made in a bacterium that you usually find inside your gut. And that was the first commercial use of recombinant DNA technology. This probably counts as the worst slide known to mankind, but it's an attempt to demonstrate what I just told you. You have a bacterium, E. coli, that has a plasmid, and we take the plasmid out. It's very easy. You just put it into some detergent and the plasmids fall out because the outside of the bacteria gets holes in it. We go to a pig and we find the gene that makes insulin, and then we combine the two, and that's called recombinant DNA. This plasmid now has a gene to make insulin. We put it into a bacterium. It now is able to make insulin. We do what's called cloning, which just means grow up. A clone is an identical copy of the cell with the exact same genetic makeup. So this divides and makes an identical copy. It divides again, divides again, divides again. They're all making clones. And then we get the product, which is, in this case, insulin product. Very simple, very classic application of genetic recombinant DNA. Everyone okay with that? Is this making any sense? Yes, thank you. This class is just too big. Another breakthrough, if you like, in technology, which is incredibly useful and popular, and you'll read about it all the time, or you should at least understand its potential, is known as the polymerase chain reaction. And this is known just by its initials, PCR, PCR. And the instrument itself is very basic. Every laboratory now, any molecular biology lab has many of these. It's simply just a small instrument, only costs a few $1,000s, that you put in little, these are little centrifuge tubes. You put the tube into this block, which is really just something that cycles hot and cold very quickly. It's called a thermal cycler. Thermal meaning heat. And so a thermal cycler is what you use to do the polymerase chain reaction. And what it does is it allows us to take a very small piece of DNA. Let's say you just take one strand of DNA, and then you put it into this thermal cycler. When you raise the temperature, those of you that know anything about DNA know that it's a double strand. You've probably heard of a double helix. It's in a helical form. And that double strand is bonded together by fairly weak bonds. If you heat it up, they come apart. And that's called melting. So if you get to about 70 degrees, the piece of double-stranded DNA that you've left, that you started with, will melt. Once it's opened up like that, by using an enzyme to elongate the new piece of DNA and to stick on complementary bases to it, you go from one piece of DNA, it melts, it forms a copy there and a copy there, and suddenly by cooling down, the two strands come together and you've got a copy of it. You've gone from 1 to 2. We do it again. These two strands melt. They form a copy of themselves. We cool down, and we got 4. You do it again, you get 8. Do it again, 16. Do it again, and you keep going. And that's called thermal cycling. Hot, cold, hot, cold. Within a few hours, you've gone from one piece of DNA to billions and billions of identical copies. Wow. Love it. So PCR, it uses that enzyme we talked about from that bacterium last time from a hot spring, because you need an enzyme that doesn't mind getting heated up to 70 degrees and melting the DNA. So DNA polymerase is the enzyme that makes the new copy of the DNA. And it can work because it doesn't mind 70 degrees. It was found in water at 70 degrees. And so Thermus aquaticus is the bacterium you'll remember, and that is the enzyme that everyone on this planet uses if they're a molecular biologist. And what that allows you to do is take a single strand of DNA from, for example, a crime scene. So someone has committed a crime and they've left traces of blood or traces of semen, depending on the nature of the crime. They could be invisible because you're talking about 1 or 2 strands of DNA. And the crime scene investigator can take that little piece of DNA that someone's left behind. It could be a skin cell, it could be anything. And you can use PCR to make billions and billions of copies of it. Once you've got those copies, you can chop it into pieces with a restriction enzyme. And that allows you to get what's called a genetic fingerprint. And that looks like something like this. So by just randomly chopping it, you can see the different way the bands come out of the pieces that you've chopped. The different sizes of all the little fragments you've made can be put down in a gel like this, and you can get a fingerprint that is absolutely unique to the person who left that DNA behind. Everyone's DNA is different. So if you were to chop it using an enzyme, it's going to chop into different size fragments, depending on who you are. And those fragments are shown on this fingerprint. So this is how we find who did the crime. This is what genetic fingerprinting is all about, that you hear about every day. If you read the newspapers, they do DNA testing to see who's the parent, to see who did the crime, to see if someone's innocent. This is what they do. They take some DNA, they do PCR, they put it on here after cutting it with enzymes, and they look at the size of your bands, which will be different from this person, who will be different from this person, different from this person. Very powerful tool. Okay, so I've got you up to speed on how you do biotechnology, whether you believe it or not. We've looked at microbes, we know something about them, we know what they give us, we know that they give us the tools, whether it's scissors or whether it's enzymes. And this has allowed us now to do fairly amazing things with biotechnology, which then brings up the whole question of ethics. And this is where you have to weigh in the future, because this is moving at a scary speed. So remember, biotechnology has been around forever. We don't have to use manipulations in the laboratory to make it work for us. We make cheese every day in life, and that uses bacterial enzymes to make the milk curdle and then give us the cheese. We, of course, use another kind of microbe called a yeast. We didn't actually include that in the first lecture, but a yeast is a single cell. And yeast can then ferment sugars to give us alcohol when we're making wine. Most of you that are sitting there wearing a cotton t-shirt, that probably came from cotton, which is now fairly universally prepared by growing cotton plants, which have been genetically engineered so that they're resistant to insect pests. So think about it, that cotton plant has had a gene put into it which will make an enzyme which will kill the insect that is trying to eat the leaf of the plant. That's how powerful genetic engineering is. And because cotton is such a valuable crop worldwide, much of the cotton that is grown now has been engineered so that those insects don't destroy the crop. We'll come back to what that's all about shortly. I told you that insulin is made 98% from bacteria now, from the bacterium Escherichia coli. There are about 2% of the population that can't use that insulin, and we don't understand why. So it's not universally used, but it is used by about 98% of all diabetics. They're using insulin from E. coli. And of course, if you go for a blood test or you go for any kind of test to the doctors, very often they take a little blood sample, they put it onto some sort of test plate, they look for some reaction like a color change, and they can diagnose almost immediately whether you've got a certain disease. And all of those rapid tests are because of biotechnology and the products we can make from biotechnology. They can genetically manipulate so that some color reaction will happen if a piece of DNA from a pathogen comes in contact with it. And many, many other applications are all happening now. Most of biotechnology, we're waiting for innovations in the future, which is why you need to pay attention to it. There's huge promise that biotechnology is going to help us replace diseased organs in the future. Maybe we can repair damaged neurons and use biotechnology to go in there and replace those cells that are damaged and fix the spinal cord. Christopher Reeves, who you know is paralyzed, an actor, has spent vast amounts of his money trying to push for a cure to spinal damage, and that's using biotechnology. There's a hope we're going to produce many new vaccines and treatments that will be much more effective. Again, we touched on it last day. Think about it. You go to the doctor now and it's not always certain they can cure you. We cannot cure most viral infections. We just have to slow down the viral attack. There is no cure for AIDS. There's no cure for the common cold. There's no cure for herpes. The list goes on. We are pretty good at killing off bacterial infections, but remember, bacteria are changing. They're getting ahead of us. They're becoming antibiotic resistant. So there's constant challenges to stay ahead of the microbes that are trying to kill us. We can produce more nutritious plants using biotechnology. That's already happening. We can produce plants that can grow in hostile environments, meaning we can start growing them in areas that we cannot normally cultivate food plants. And we're looking at alternative energy as another way of adopting biotechnology. The great hope here is, and a lot of people are working on it, that we can use these algae, these single-celled algae, which often have oil droplets in them. You manufacture, using biotechnology, a type of alga that produces a large amount of oil, and then you can start growing that, in cultivating it in sunshine, and growing these products in a large fermenter that uses sunlight. We do this already, but there's hope that we can really do it to produce oil from single-celled algae. And that way we can move away from having to use hydrocarbons from petroleum. So all these things are being done at the moment. The question is how we're going to scale them all up. So you should all be concerned about, what's the way we want to live and deal with this. And that gets us into this great topic of ethics. Ethics is, by definition, the science and philosophy of morality. So you need to be aware of this. You all have to make decisions yourself as these innovations come about. We tend to, when we're talking about anything in the biology field, we tend to now coin bioethics to be a little more specific. And that, of course, it was coined in 1971, but now has broadened to include anything in biology, biotechnology, medicine, population control, environment, and ecology. So bioethics is just a little bit more specific to the subject we're talking about in this class. Of course, ethics is just how you make decisions in your life, so it doesn't have to be about bioethics. We all have an ethical clock within ourselves. We just have to be in tune with it. So let's think of some of the the challenges out there to what you are going to be challenged with in the next 10, 20 years. And the first one, of course, is the outcome of the Human Genome Project. This was a project that was started 13 years ago. It's finished now. And it was a worldwide effort to understand what the human genome is made up of. So your entire DNA sequence is made up of bases, and it's the order of those bases that determine where your genes are. And there are only 4 bases in nature. I will not even give you the names of them. Some of you do know if you've done biology, but the bases are A, T, C, and G. So in other words, the unraveling of the human genome was simply a question of sequencing the entire human genome to find the sequence of bases. Sounds fairly straightforward. Of course, it was a monumental effort, and it's the sequence of those bases in a segment called a gene that determine what that product is that's made from that gene. As a result of the Human Genome Project, which is now published and is public information, we have identified 30,000 genes in the body, so we know where those 30,000 proteins are manufactured. Your genome is made up of much, much more DNA than is contained in just those 30,000 genes. We have a lot of junk DNA in our bodies, that is DNA sequences that don't code for a gene. We have been subject to incredible amounts of viral attack in the past, and a lot of junk DNA has been incorporated into our genome. So the genome is a massive amount of DNA, and the sensible parts contain the 30,000 genes, but you have considerably more junk DNA, all of it different, which of course, if you think about it, is why genetic fingerprinting works, because all the junk DNA in all your bodies is different, and so when you chop it randomly, it all comes down as different sizes. If you had all the same genes, then everyone would be chopped the same. Think about it. Knowing what the Genome Project can do for us, John Kornfeld, when he was introducing me, said that we've now got the potential to say, well, congratulations, Miss Jones, you have the gene for breast cancer. That is not particularly useful unless you can do something about it. And so knowing that someone has a gene for cancer is one piece of information. Knowing how to use it is the ethical part. You would not want all the medical insurance companies to know that certain patients were susceptible to cancer in the future. And so that information has to be used in a very guarded way and in a very legal and ethical way. The big piece of molecular biology that we haven't really come to understand is how to treat those conditions. So it's all very well recognizing that you have a gene for cancer. It's only useful if we can turn it off. And that is where most of the research is right now. And it will come very quickly. We will have the ability to turn genes on and to turn them off, so that if we find that a patient has got a gene for a cancer, the treatment will be turning it off. And that's where most of the research is going at the moment. We have a lot of controls at the moment on what we do with the information about the Human Genome Project, and much of that is due to the work of James Watson, who started an organization called the Ethical, Legal, and Social Implications Research Program. You don't need to remember that one. Just be aware that James Watson, who was, of course, one of the people who obtained a Nobel laureate for unraveling the structure of DNA. You'll remember Watson and Crick, absolutely, right? So James Watson has continued researching and was one of the lead researchers in the Human Genome Project. But he realized the need to set up ethical controls, and that's what he did, and it still exists to this day. There are many organizations watching the ethical ramifications of this project. We'll move on. I'll skip something. Another one that's hotly debated, and you have to weigh in as well, is whether we should do animal cloning. We have the ability to do animal cloning right now, and we do it. A lot of animals are cloned for experimental research. At this point, we have never cloned a human, but of course, lots of people have the capability to do human cloning. A clone, as you remember, is an exact genetic copy. We talked about a clone when we made our recombinant bacteria, E. coli, divided it. Those are clones. We can make an exact copy of a multicellular organism in a similar way by genetically copying the parent. There's two ways to do this. The first is straightforward. It's called twinning, and that's where you split the embryo. And that's what happens naturally. If you have an identical twin, this has happened in the mother's womb. The embryo at a very early stage has split, and then it makes two identical copies, and that is known as twinning. And that can be done, but that's not really what we mean when we do animal cloning. The real interesting part is to do something somatic cell nuclear transfer. So in this case, and this is the real potential of cloning, you have to take an embryo cell and take the nucleus out. So that embryo cell has the ability to grow into an adult. But if you get it at a very early stage, it's not yet started to reproduce its cells, and you can take the nucleus out. Fairly straightforward. You just do it with a very fine glass needle. You then take a nucleus from the organism you want to copy. So if you want to copy someone in the front row here, you take a cell out of their skin, take the nucleus, and put it into that embryo. And then let that embryo go back into a donor, implanted in a surrogate mother, and that will grow into an identical copy of the person in the front row here. That's somatic cell nuclear transfer. Somatic cell meaning a surface cell into an embryo, and then you make an identical copy of it. We could do this with humans, but we're not allowed to. Not to make adult copies anyway. And so the success is, it's hard work. It doesn't always work. The very first one, I think most of you have probably heard of Dolly the sheep. If you haven't, these are good things to Google. And that was the first example of animal cloning ever done. And it was to make an identical copy of a sheep that was called Dolly. But that was the success rate. It took 277 embryos before one actually grew into an adult sheep. We can do it with cats, and there the success rate is about 1 in 87. With cows, for some reason, it's very successful. It's quite easy to clone a cow. About 1 in 3 embryos will take. But again, we've not yet cloned a human adult, and that's where the ethics comes into it. That's where you all have to be thinking. Why would you want to do it? Well, you might want to produce identical children. Not a great idea, perhaps. I'm leaving that up to you to make that ethical decision. Whether as a future parent, you might want to make an identical copy of a child, perhaps, that has died in the past, or you just like your first child so much, you want to have an identical copy of that child. And you pay money and you make an adult, you make a clone of them. That obviously raises some pretty ethical questions. We do therapeutic cloning at the moment, and that's to make embryos, human embryos, for something called stem cell research. And this is something that you really have to pay attention to. A stem cell is one of those cells that is at a very early stage in the embryo that can, once it's undergone the right set of instructions, can make itself into any cell in your body. So that cell early on in the embryo, those cells are called undifferentiated cells. They haven't yet decided whether they want to be a liver cell or whether they want to be a skin cell or whether they want to be a stomach cell. They could be any of those, but the embryo is too young to have started to differentiate into organs and skin and other components of the adult. So if we clone those cells very early on, we can use them in biomedical research. So we can make those embryos, implant them into the womb, or you don't have to implant them, you just have to grow them in culture, let them develop, and then you can harvest them, because those stem cells have got the potential to be any cell in the body. And that's where the real promise is coming from. You can get adult stem cells, because of course if your tissues get destroyed, they can regenerate and in an adult repair mechanism, and those are from adult stem cells, which are much less controversial, but they don't work anything like as well as embryonic stem cells, which have got this full potential to be anything you want them to be. We're quite worried about stem cell research. Some people think it's a good thing, people think it's a bad thing. There was a resolution by the United Nations in 2005. It's called a non-binding resolution. The UN cannot make world rules. It can only make suggestions that we all follow them, so they're non-binding. But it basically called on all the governments to ban all forms of human cloning. They thought it was important from an ethical standpoint that it didn't happen. 87 countries were for the ban, 34 countries were against it, and 37 abstained and didn't vote. So in other words, this clearly showed that across the globe, countries prefer to make their own policies on therapeutic cloning. In America, it's a split. About half the states in America don't allow therapeutic cloning. About half do. California is one of those states that does allow it. We do a lot of therapeutic stem cell research here, but you are not allowed to use federal funds. So they cannot apply to the National Institute of Health, for instance, to get money to do stem cell research, but a private donor or a private foundation can fund it, and California is taking the lead in stem cell research. So just to recap, these are cells in multicellular organisms that can differentiate into any needed cells. They come in two forms: embryonic ones, which we can get by doing the early stages of cloning, and adult stem cells, which are less interesting because they're not so flexible. And of course, the promise here is that you can grow new organs, you can repair damaged tissue, and you can repair spinal cords and many other things. So this is the promise. You take those cells, which are at a very early stage, you put them into the spinal cord, you give it the right trigger to make it make new nerve cells, and suddenly you've got a cure for spinal cord injuries. That's where the research is going. We're not there yet. The controversial part is it means that you're taking human embryos that you have made in the laboratory, and you grow them for 4 to 7 days, and then you harvest them. You can see that in some people's minds, this is a very controversial topic, and in other people's minds, it's less so. You have to make up your mind about stem cell research. And many people know what a GMO is. Please, at least one or two. Thank you. Genetically modified organisms. I think this is a good example of we need to make sure that everyone, certainly at Sonoma State, and hopefully all young people on the planet, start to really become a little bit more conversant with the potential changes in science. You all need to know what GMOs are. You just voted on it in a California proposition. It just completely floors me that that proposition didn't pass. You were being asked to vote on whether you wanted food to be labeled as GMO, yes or no. California population said no for some reason. Maybe it was because people didn't know what a GMO was, or maybe people didn't want to know what their food was, how their food was made. But to me, I think it's important that everyone here knows what a genetically modified organism is, because you're eating them every day. So think about it. If you do some modification to the gene of a corn plant or a cotton plant or something edible, you've genetically modified it. And often it's to add genes that make it grow faster, to make more food, to resist herbicides, to resist pests. They are now making plants that you are going to eat with a gene that makes that plant. That's great food. You then eat that plant that is being covered in Roundup. Is that safe? You have to decide. It probably is if you wash it well, because that's on the surface. A little bit more worrying for me is, think about a plant that resists pests. So in other words, somebody has manufactured that plant, put a gene into it, which now makes a chemical that kills a pest that is trying to eat it. And they're telling you that that new chemical that kills a caterpillar is safe. You have to decide whether you believe that company. Maybe that chemical gives you cancer and you don't see the cancer for 50 years. Or maybe that chemical is totally safe. Those are the decisions you have to decide, whether you want to eat food that has been genetically modified. Companies that make these plants are huge, the biggest being Monsanto. They spent $63 million lobbying to make sure that crops like the one that just failed in California will prevent labeling of food. That tells you how much profit they're making, how committed they are to making sure you don't know what you're eating. Is it safe? They say it is, and I'm not in a position to say it isn't. That is your ethical decision. There are 3 agencies watching it, and the Food and Drug, the Environmental Protection, and the U.S. Department of Agriculture. At this point, they are telling us it's safe, but you have to make the decision. So, I'm going to stop there and now give you a video, which I'm sure you'll all be thrilled about, because you don't have to listen to me. But I hope this is making sense. Microbiology, the phenomenal tools from microbiology, the ability to manipulate in laboratory and do all sorts of amazing things like tell an E. coli to don't bother living in someone's gut, go grow in a fermenter and make us some insulin, or put a gene into food to make it a GMO, or do some amazing things with stem cell research if you believe in human cloning, therapeutic human cloning. These are all the little pieces that will be reinforced in this 20-minute video on bioethics, and so use this now to reinforce what I've been trying to get across to you.
Biotechnology, the use of biological organisms and processes to provide useful products in industry and medicine. It has begun to affect so many aspects of our 21st century lives. Biotechnology applications can be seen in agriculture, pharmaceutical development, chemical engineering, and even the human genome. The use of this technology expands and grows each day. But what exactly is biotechnology? How does it affect everyday life? Where will it take us in the future? Biotechnology in the 21st century will help demystify this seemingly limitless and vital science by providing concrete answers to these important questions. Throughout history, powerful technologies have advanced so quickly that little thought was given to their impact on society. While the nuclear age remains a bitter memory, how will the emergence of biotechnology affect our future? Will it change life as we know it, for better or for worse? In the biotechnology field, bioethics is the study of ethical and moral implications of new biological discoveries and biomedical advances.
Well, medical ethics is important to society today, primarily because there are so many new ethical dilemmas that have arisen in medicine as a result of changes in medical technology. Essentially, what the technology does is it alters the kinds of choices that we can make as members of a profession or as members of a society. And some of the choices that become available to us now are choices that will challenge what had been fairly standard kinds of moral understandings of what seems right and wrong and what's reasonable or what's unreasonable.
But what are the moral and ethical implications of using science to alter nature's creations? And is it our duty as citizens to decide how biotechnology should or should not be used?
There are several major oppositions to biotechnology. The first one that usually comes to mind for a lot of people is the issue of safety. One of the tricky aspects of figuring how safe any technology is being able to predict correctly what the likely consequences are of that technology. Given science today, we can usually predict with a fair degree of reliability the immediate safety issues associated with an emerging technology. We can do that because we know the environment in which it's going to be deployed and what the likely interactions are in this current environment.
It's a very complex environment in which the biotechnology plays. It's the hearth in the home, or you're dealing with someone's life. So you have to be able to assure yourself as a company or assure your customers that what you're giving them has been tested and to the best of your ability proven safe.
When we talk about morality, the way in which at least philosophers use that word is to refer to what I'll call prevailing ethical beliefs and judgments for one or another social group in our society. Some of those moral judgments, traditional moral judgments, may be quite reasonable and defensible and sort of worthy of continued respect. In other cases, we may have to adjust our moral practices. And this is where ethics then comes in.
The good news is there's a lot of mechanisms in place for clinical and translational and basic research that help protect us to do the right thing and to do ethical experiments so that, the use of animals, the use of stem cells, and using the right stem cell lines.
We're harnessing something that's inanimate, that doesn't have a role or a decision in this, to make people's lives better and to help people. And if we really focus on that, I think the ethics of biotechnology are clear. I think it's when people look at misuse of biotechnology, biotechnology being used in war, in weapons, and essentially I don't think anyone who's legitimately involved in the biotechnology industry or movement embraces any of that. So I think if we stick to the central focus of what we're doing, it's pretty clear that we're trying to do good.
Who do you make the watchdog over this? That's the question. And I think the mistrust comes when things are done in secret and the controls are not clearly stated. And honestly, I would be concerned too if those two safeguards weren't in place. So I think one thing that the scientific community can do is to really be transparent, to not try to do things in secret.
In an effort to make biotechnology an accepted part of society and to prove that their goals have good intentions, scientists are taking public opinion and ethics into consideration when developing new technologies. Scientific advancement versus the dignity of human life is an issue that reaches the highest level of debate. Since the world's first cloned animal was born in 1996, speculations of taking this technology and building a better human has many people concerned.
When you apply biotechnology to animal breeding, you can sometimes get into cloning. This is a difficult area because cloning, which is to create a replica organism based on the genetic material of the donor, can be controversial and needs to be looked at very carefully. Sometimes it could be an advantage. Cloning bacteria, for example, has had great advantages in developing antibiotic drugs, developing insulin, and the like. But cloning higher organisms like people or sheep or cows, that's something we have to look at very carefully and look at the benefits and look at the risks.
With people talking about cloning animals, is that right? Is that wrong? It's your core ethic and understanding whether it is right or wrong in your own belief. Unless there is some burning need because a strain of animals has been made that produce a specific drug that could save a mega portion of the people, I would say cloning an animal for any other purpose is actually rather selfish.
We do cloning at so many stages and for so many different applications, and most of it is completely, everybody would consider completely harmless. But then of course everybody thinks of Jurassic Park or of making the perfect human being, and those are areas where people are bothered, and completely rightfully so. So I do understand that, but often it is just our limited view or exposure to the reasons why people are doing it. If we can prevent an illness, if we can prevent a disease, why shouldn't we try to use this new approach if it involves cloning and prevent it?
One of the things that we try to do is when we are looking for answers that we want to apply universally, we try to eliminate as many of the variables as we can, and cloning allows us to do that. If we can take a single cell with a single genetic code and reproduce that, as we treat that cell and as we manipulate it, we can be fairly certain that the answers that we're getting are not because of genetic variability, but because of the therapy that we're applying.
Technically, the ability to clone humans is within the reasonableness of the technology. The ethics surrounding that are much debated, and they have to be debated. We're talking about the ability to create a sentient, conscious life form, and that should not be taken lightly. I think perhaps the benefit of being able to clone a human would be not necessarily in being able to generate an entire organism, but rather to be able to control the development and organ regeneration so that you can have autologous donations to yourself. If you needed a liver transplant, for example, that well in advance of your surgery, they could clone yours and grow a liver that had the same genetic background and the proper major histocompatibility complexes, so that you wouldn't have to be on immune suppressing drugs the rest of your life. So you could take forward transplant biology significantly.
Education is probably the best way of handling it, so that people really understand what is being done, what is going on.
The real question we want to ask, I think, is how cloning might be used or could be used in the future. The fear that was, sort of initially spread when Dolly was cloned was that there would be these mad dictators in the world who would create 1,000 or 1,000,000 Hitlers or other clones of themselves. That's just science fiction. That has nothing to do with the real world.
Is it right or wrong to clone basically a human life? Honestly, I don't have an answer as far as yes or no, whether it's right or wrong. To clone human life for commercial gain, I think there's a consensus that would be a bad thing. This is one of those complex questions that there's no easy answer to. I think complex questions require complex answers.
You have to be very, very careful how we proceed down that path, because there are many, many ethical and moral considerations. You don't create life to destroy it. And when you are cloning a human, you are creating a life form, and it's just because it's a duplicate doesn't mean that it's disposable.
As of 2008, no one has been able to clone a human being or prove that such a thing is even possible. But what seems like science fiction right now will be possible in the future. It is likely that the world will one day see some form of a human clone. All the cells in our bodies begin with an original fertilized egg called the zygote. This zygote continues to divide, generating more and more cells, and those cells become the tissues that form a human fetus. Some cells at this early stage have the ability to become any type of cell in the body. These cells are known as embryonic stem cells.
Embryonic stem cells is when you actually take an embryo and break it apart and collect its cells. And each one of those cells has potential to become an entire person. If you take out some or all of the genetic material and implant new genetic material, then you can control what kind of person that person becomes. If it's a human embryo, if it's a sheep, what kind of sheep that sheep becomes.
As the field of stem cell research progresses, scientists are discovering ways to harvest stem cells without having to use embryonic material.
The ethical concerns behind stem cells are not really behind the technology. I think the ethical concerns have come from the source of the stem cells. And there's been a debate about that because some people have been gaining stem cells from embryos. Fortunately, it looks like we can now develop stem cells from multiple sources, even from the individual themselves. And those types of advances, I think, are going to remove the ethical issues and allow us to really focus on the technology challenges involved in stem cells.
We no longer need to rely on fetal material for stem cells. We can isolate them from our own bodies. That field is beginning to expand and explode. So the sources of stem cells are the issue, not necessarily the use of stem cells for most people, I believe. And if we can educate about the sources of stem cells and move away from where people are anchored, I think that there will be a greater acceptance of the utility of very beneficial technology.
The other type of stem cell is stem cells that are collected from other tissues in the body. One source is umbilical cord blood from a newborn. And what we're finding is that many tissues in the human body have stem-like cells. There are stem cells in the brain, stem cells in the skin. These cells can't become anything, but they can become a lot of things.
And so the hope is, under regenerative medicine, that we'd be able to use our capacity to manipulate those stem cells for purposes of creating tissue that would repair damage in one or another part of our body. In the case of individuals with diabetes, the goal would be to use stem cells to create the islet cells that would in effect correct for the deficiencies that diabetics are faced with, or in the case of spinal cord injuries, to create nerve tissue that would repair a severed spinal cord.
I think that the real potential comes in unlocking the cells that we currently have available to us. In other words, if we can't take a muscle stem cell and regenerate muscle, I don't think we're going to take an embryonic stem cell and regenerate muscle. That science is a lot further along than you might think, but nowhere near the completion. So yeah, I have a lot of hope that this science is going to produce real therapies in the near future. We'll see.
In order to ensure that new treatments created by biotechnology are safe for use in humans, they must go through a battery of tests. Animal testing is considered a necessary part of researching and testing new drugs.
The unfortunate reality of the drug development business is that you have to do animal testing. There is no ethical way in which you can take a new chemical and put it into a human until you start to define the toxicities, the doses that can be tolerated by an organism, a model organism.
The use of animals in research is absolutely fundamental. What we do a lot and where bioinformatics comes in is, we often use mice, sometimes rats, as surrogates for the human. We can measure the whole genome of a mouse, and we can measure the whole genome of a human, and we can do a comparison between the two to see the similarities and the differences between pathways, and it helps us understand how working with laboratory animals can inform the work that we do with humans. So, laboratory animals are very important for biomedical research.
The state of biology is advancing and pharmaceutical science is advancing to where we can do many of these things in petri dishes or test tubes. The complexity of an organism will never be replicated in a test tube. There are just too many nuances and varieties that you have to go through into a test organism.
All life is sacred. So we've been working very hard to find model organisms and develop these organisms like mice and rats and fish and sometimes even primates that can help us understand, very dangerous diseases, can help us understand the mechanisms of disease. And, we work very closely with the government on this. We partner with academic research groups and other groups that are concerned with the ethical treatment of animals and research to make sure that we're applying the highest standards and, we're doing the right thing.
In the development of drugs, I don't foresee in the near future the ability to avoid animal testing. The complexity of an organism requires that we test in animals. We're not going to be able to reproduce that in a test tube in the very near future.
While animal testing is essential to the development of new drugs, researchers are looking for new ways to screen drugs that will lessen the dependence on animal test subjects.
There are steps being made. I know of a burgeoning new service organization sector in biotech that looks at toxicity in the test tube and helps companies select only the most promising drugs. So they're able to eliminate drugs before they have to go to animal testing. Thus, we reduce the number of animal tests that have to be performed by making better selection of drugs and eliminating those that would not make it past animal testing successfully. So we'll not be able to eliminate animal testing in the near future, but we can certainly reduce screening of new drugs in there to only the best candidates.
Biotechnology is likely to become one of the most powerful emerging technologies on the planet. People are passionate about its possibilities, but virtually everyone close to the technology agrees that scientists and corporations must use it to better our world, not harm it. As citizens of the global community, we need to decide how biotechnology should be used.
I think society has to create a standard, what we call ethical and what we do not call ethical. I think there has to be a general agreement what is acceptable
We have to avoid being, I think, excessively cautious and fearful about new technology, but we also have to avoid being excessively ambitious. We have, from Greek mythology, the story of Icarus flying too close to the sun. We do fine with airplanes. They seem to get us where we're going safely and quickly. But there are other kinds of technologies whose consequences need to be assessed before those technologies are very widely disseminated.
We're often fearful of things that we don't really understand. It's just human nature to fear something that we don't understand. And the challenge of biotechnology is that it's difficult to understand if we don't take the time to try to understand it. It's as simple as that. The good news is that, especially with the internet, we can understand biotechnology. Biotechnology, like most things, involve benefits and risks. And so what we need to do is look at the benefits and risks and see and make a measurement. Look at the balance and see whether the benefits outweigh the risks. And it's something that we have to do together as a society.
Consumers should watch with biotechnology exactly what they're getting. The companies really need to inform the consumer, this is how we did it, this is why we did it, and this could be the potential downfall if it is something that could harm us. But they need to be able to educate the consumer so we can make a choice.
As we start down to develop more and more targeted and better refined drugs to treat disease, we're going to happen upon many ethical discussions we have to have. And we have to have these discussions as a society, as a group of healthcare providers, as companies. There's a litany of moral and ethical questions that no matter where you work in the biotechnology space, you're going to have to grapple with. And it's my opinion that you're better off having those conversations in an open fashion with consumer advocacy groups, bioethicists, physicians, and ultimately come to an acceptable solution. Because biotechnology is here and it's here to stay.
How come I don't get a clap? Okay. You're all dismissed. That might get me applause. See you Wednesday.